Hello everyone, this is Dr. Priyanka Sajdev here. In this session, I will be discussing the various types of staining techniques in microbiology. Now, to see the microorganism, we require to stain them. So, let's discuss the various types of staining techniques available in microbiology. So, here I will discuss first unstained preparation, also known as wet preparation. First, I will discuss their importance, and then I will discuss what is the requirement of staining in microbiology and what are the different types of stains available in microbiology. So, I will be discussing simple stains, negative stain, impregnation staining or methods, and the most important differential stains, that is gram stain, acid-fast stain, Albert stain. In the end, we will see a special type of staining technique, that is bipolar stain. So, in this order, we will discuss all types of stain. Let's start the various types of staining techniques. So, to study the bacteria, especially bacteria, we have two types of preparations. The unstained preparation, known as wet preparation, because we can use it only when they are wet in the drop. Once they will dry, the bacteria will be dead and it is not visible. So, the unstained preparation has to be observed in wet state. Not in dry state. And the second type is the stain preparation. Now let's start with unstained preparation first. Unstained preparation, we cannot see the morphology of the bacteria. Now you have to understand this. All bacteria are transparent. They do not have any color. They are transparent. And the slide on which we put the bacteria, that is also transparent. Human eyes cannot see a transparent thing on a transparent background. If you want to see anything, you require contrast. Now, in the same way, if you want to see the bacteria which are transparent, you have to stain either the bacteria or you have to stain the background, right? So, if you use the unstained technique, you cannot see the morphology of the bacteria. Then why at all we use the unstained preparation? The unstained preparation is used to see the motility of the bacteria, not the morphology. Please mind my words. I am repeating again, the unstained preparations are used to see the motility of the bacteria, not the morphology of the bacteria. We cannot see the size, shape of the bacteria, the structure of the bacteria on unstained preparation. But on unstained preparation, we can see the motility, right? Imagine a transparent bacteria is moving on a transparent background, that is slide. So we can see that something is moving, but we cannot make out its exact structure, its shape and the morphology. But we can make out that something is moving. So that's why the unstained preparations are used to see the motility of the bacteria, or else these are used for very thin bacteria. Which is the thinnest bacteria in the world? The thinnest bacteria are the spirochetes. They are thread-like. They are as thin as a thread. So, for the thin bacteria, the spirochetes in dark ground microscopy, they are used. These are the only two used of unstained preparation. Now, how to make an unstained preparation? What are the steps? So, you have to take a cover slip. In this image, can you see this is a cover slip? You have to take a cover slip with the help of a toothpick. With the help of a tip of a toothpick, you have to place the Vaseline at the four corners of the cover slip. At the four corners of the cover slip, you have to put small Vaseline, right? Now, you have to take the inoculation loop. In the inoculation loop, you have to put your inoculum at the center of the cover slip. It's a liquid drop. It's a liquid culture, bacterial culture, the inoculum at the center of the cover slip. You can see this is the drop. It contains bacteria. And at the four corners, the Vaseline is there just to provide the elevation. Now, what is the next step? What is the next step here? In the next step, you have to take a slide. This is a slide with depression at the center. It's not a flat. There is a depression in the center of the slide. So, you have to put that depression just above the drop. Can you see? I'm putting this depressed area just above the drop. So, this is the cover slip. On the cover slip, this is the drop at the center. This is the drop at the center. At the four corners, there is Vaseline. So, I'm taking a slide with a depression at the center. So, I'm putting the depressed portion just over the drop and like this, and then I will invert it. Now, you can imagine if I'm inverting the drop of the bacterial culture which is present at the center of the cover slip, if I flip-flap, if I invert it, the drop will hang in the air. That's why the wet preparation, the unstained preparation is known as hanging drop preparation. You got my point? This is the reason why they are known as hanging drop preparation. So, you can see the same thing is shown to you. First, you have to put the depressed area over the drop, right? So, depressed areas over the drop and the Vaseline is just to provide the elevation, a little bit elevation. And you have to stick the Vaseline here, and after that you have to quickly invert the depressed slide. You have to do the flip flap like this. Invert the depressed slide. Drop will now suspended from the cover glass inside the concave hollow in the slide. So this is the drop which is hanging in the hollow of the depressed slide. You can see a beautiful diagram here. Let me show you. Can you see this is the slide with the depression at the center, please? Can you see this is a slide with the depression at the center? Yes. Can you see this is the cover slip? Yes, you can see. Can you see this is the Vaseline just to provide the elevation? Yes. And what is this? What is this? The center here. So it's the drop. It's the drop which is hanging in the hollow cavity, in the hollow depression. Now you have to quickly examine it under microscope. Why I'm using the word quickly? Once the drop will get dry now, the bacteria inside that will be dying. They will be dead. You cannot see anything. If you want to see the motility of the bacteria, you have to observe it when it is wet. So it is known as wet preparation. So unstained preparation are basically wet preparation. They are known as wet preparation, and they are also known as hanging drop preparation. Now you know the reason behind it, right? So you can see the drop is suspended. Now you have to observe it under microscope. You can see this is the slide. We are observing it at 10x. At 10x, you have to observe it and start from the edge of the hanging drop. From the edge, don't start from the center. You have to start from the edge towards the center. So there are chances you can see the motility of the bacteria. You can see only the motility. So only motile bacteria can be seen here. The non-motile bacteria cannot be seen on hanging drop preparation. Please mind it. You got my point? So this is the thing. Can you see here? Can you appreciate the motility? Now, if I ask you what is the shape of the bacteria, what is the size of the bacteria, what is the structure of the bacteria, can you answer? You can't. You can't. So you cannot comment on the morphology of the bacteria on an unstained preparation. But yes, on unstained preparation, you can comment on the motility. I can see something is moving, but exactly what is moving, I cannot comment on that. So the unstained preparation is for motility of the bacteria. It's for motility. It's for motility of the bacteria. You got it? Can we go ahead? Yes, we should. So that's all about the unstained preparation. You should know the steps, you should know the utility. The utility is only one, motility. But what if we want to see the morphology of the bacteria? What if we want to see the shape, size and structure of the bacteria? We have to do the staining because of lack of contrast. The reason behind staining is the lack of the contrast. I told you bacteria is transparent, the slide is also transparent. We cannot see transparent on a transparent background. We have to provide the contrast. We have to. So what are the different types of techniques available? There are 4 types of staining techniques available in microbiology. Let me read their names. The first one is the simple stain. The second one is the negative stain. The third one is the impregnation stain. And the last one is the differential stain. Let me start them one by one. You should know their definition along with their example, along with their image. Now, in various competitive exam, it's a trend. You get 25 to 30% of the paper as IBQ, image-based question. And microbiology is one of the vital subject from which you can get many image-based questions. Microbiology is all about images, you know. So wherever there is an image, you should understand that image. Instead of learning it, you have to understand the concept behind that image. So here I will show you the various images of various staining techniques. You have to learn that image also, right? So the first one is the simple stain. Why simple stain is known as simple? What is the meaning of the simple? Simple means it gives same color to all bacteria. All bacteria are uniform. Like the school child, they are in uniform. They all are same color. Can you see the slide? All bacteria in same color. I can see all bacteria are blue color. They are all blue. There is no different types of color. That's why known as simple stain. There are 2 examples of simple stain you have to learn. Methylene blue, basic fustian. What I am saying? It's methylene blue, number 1, and basic fustian, number 2. So these are the 2 examples of simple stain. And learn the diagram. Coming on the second type of stain, it is known as negative stain. What do you mean by negative? Have you seen the hard copy of the photographs, the old photographs? So along with the photographs, there are negatives. Have you seen the negative of a photograph, the hard copy? What do you mean by negative? Negative don't stain the structure to be seen. Negative stain the background. I told you the bacteria is also transparent, the background is also transparent. In the last stain, the simple stain, it was staining the bacteria, giving same color to all bacteria but not the background. But here, instead of staining the bacteria, we stain the background. So the ultimate purpose is solved now. We want to provide the contrast. Now either we provide contrast by staining the structure to be seen or staining the background. Right. So here we are providing the contrast by staining the background. Yes. So it is the negative stain. So here the background is stained while the structure to be seen is not stained. So can you see here? Here you can see this is the bacteria. It is not stained. You can see the background is stained. You can see. It is a spirochetes, the thread-like bacteria, the thin bacteria, the thread-like bacteria. They are not stained. They are white. They are transparent. But the background is black. In the same way, you can see this is a capsulated bacteria. You can see the diplococci, the capsulated bacteria. It is not stained, it is transparent, but the background is stained. Now learn the examples. The examples are more important. The two examples you have to learn here is India ink and Negrosan. Never forget, it's ultra super duper important PYQ, right? Number one is India ink and number two Negrosan. These are the two examples of the negative stain. Now learn the image. You can get a straightforward IBQ. If such a black image is coming in front of you in your exam with something transparent on it, it's negative stain. Now learn the two examples, India ink and Negrosan. That is the second type of stain. Coming on the third type of the stain. What is the third type of the stain? It's impregnation. What do you mean by the term impregnation? The dictionary meaning of impregnation. Impregnation meaning layering, layer over layer. It's layering, layering. It's layer over layer. It is known as impregnation. Now sometimes bacteria are very thin. They are too thin. Can you see this bacteria here? Let me show you. This bacteria is a very thin bacteria. It cannot be seen normally. So what we are doing the thickening of the bacteria by providing a layer over layer. It is known as impregnation. That is the meaning of the impregnation, right? Now we do the impregnation with silver. Silver, the chemical formula for silver is Ag. That is silver. Have you seen the sweets? On the sweets there is a coating of the silver in the same way. So here we are doing the coating or the layering of the silver, right? So here we use silver for the impregnation for layering. Now name the bacteria which are too thin. I told you that is for thin, for making it thick. So the thinnest bacteria are of course spirochetes. So the best it is used to demonstrate the spirochetes, number one. And number two, some bacteria have flagella. Can you see? Here in this diagram, these are the bacteria. Don't see the bacteria. Bacteria are not very thin. They are visible. But please concentrate on the flagella of the bacteria. Can you see the flagella? So flagella are very thin. To demonstrate them, we can use the stain. So bacterial flagella. So spirochetes and the bacterial flagella. These are the two uses of the impregnation method. Now in impregnation, what all MCQs you can get? Here you will not get an image-based question, right? So you can get the principle. For thin bacteria, we can use it. You can get a question on the silver. Impregnation is done by the silver. It's layering. And the third question you can get on the importance. What is the importance? So it is used for demonstration of the bacterial flagella and the demonstration of the spirochetes. That is a third type of stain. So till now we have seen the three types of staining techniques. The simple stains which provide same color to all bacteria. Let me write the definition. Right. Same color to all bacteria. Negative stain. It stains the background but not the structure to be demonstrated, not the bacteria. And the third is impregnation. Impregnation means layering, layer over layer, when the bacteria is too thin, right? Now, can you tell me examples of each of them? For simple stain, I told you two examples, methylene blue, basic fission, right? For negative stain also, I told you two examples, the India ink and Negrosan, right? And for impregnation also, I had given you one example. It's silver, right? So these are all important questions based on the staining of these three types. Now coming on the most important, that is differential stain. Let's start differential stain. Why it is known as differential? You know, the term itself is indicating its meaning. What do you mean by differential? Differential means just opposite of simple. What simple was? Giving same color to all bacteria. So here it's giving different color to different bacteria. Different color to different bacteria. That's why the meaning is differential. Different color to different bacteria. So here there are three examples. What are the three examples? It's Gram stain, acid fast stain and Albert stain. So we will take all these one by one in detail. You get many MCQs based on these stains. So let's start with the first one, Gram stain. So Gram stain is the stain which is most widely used in entire bacteriology. The most widely stained. So we classify bacteria based on gram stain, the gram positive, the gram negative. It is the most widely used stain. Why it is known as gram stain? Because it is discovered by a scientist. The name of the scientist is Christian Gram. In 1884 it was discovered. That's why, to give the honor to the scientist who discovered it, it's known as gram stain. That is the introduction. Now can you tell me the steps? There are 4 steps. 1, 2, 3, 4. What are the 4 steps? First you have to add the primary stain. The primary stain, which is a violet stain. Any violet stain. The gentian violet, crystal violet, any violet, but it should be a violet stain. That is primary stain, which is equivalent to any violet. The second step is the addition of mordant. What do you mean by mordant? Mordant is something which fixes the primary stain inside the bacteria, right? So I told you all bacteria are transparent now. So this is a transparent bacteria and you are giving a violet stain to this. So the bacteria will become violet from inside because it was a transparent bacteria and you provided the primary stain, but it will leak it out. It will leak it out. Now you want to fix the primary stain inside the bacteria only. So you have to provide a mordant. You have to provide here a mordant. So if you give iodine, so iodine will form the complex with the primary stain and fix the primary stain inside the bacteria only. That is the meaning of the mordant, right? It forms a complex with the primary stain inside the bacteria only. So that is the thing. Now the third is the decolorizer. Here as a decolorizer, we use either acetone or alcohol. In the alcohol, we use ethanol. We never use acid in gram stain. In contrast to acid-fast stain, where we use acid, the sulfuric acid 20% H2SO4 as a decolorizer in acid-fast stain. Here we don't use acid. We use either acetone or we use ethanol, alcohol. And the last step is the counter stain or secondary stain. The counter stain or secondary stain is any red stain. Any red will work. For example, carbol, fuchsin, saffronin, any red. So these are the 4 steps. The violet stain, which is a primary stain, followed by mordant, which is a fixer of the primary stain. Then we give decolorizer, which is never acid. It can be acetone, it can be alcohol. And the last is any red stain, which is a counter stain or secondary stain. Now, what will happen in the end? There are 2 possibilities. There are some bacteria which will retain the primary stain and remain violet in the end. These are known as gram-positive bacteria. And there are some bacteria which take the secondary stain, so they will be red in the end. These are known as gram-negative bacteria, right? Now, intentionally, I have written all this in violet and intentionally, I have written all this in red. You can see the color, the violet and the red. In the end, the violet one are gram-positive, the red one are gram-negative. Now, you may be thinking, why? Why? Why there are 2 different types of bacteria? Some are taking the primary stain and some are taking the secondary stain. So let me show you the principle also here. Can you see a beautiful diagram here? Let me explain this diagram to you. Let me explain this to you. You can see here the bacteria are transparent. You can see the bacteria are transparent here also. We know all bacteria are transparent. So on the both side we can see transparent bacteria. Now all bacteria are transparent and you are providing a violet stain. The primary stain, which is violet stain. So this on taking primary stain, it will become violet, and this also on taking primary stain, it will also become violet, right? After that you are providing mordant, which is iodine. It fixes the primary stain. So here also the bacteria are violet even after adding mordant, and here also they are violet even after adding mordant. The third step is addition of decolorizer, which makes the difference. Decolorizer makes the difference. It can be alcohol or acetone, but not acid. Now, there are some bacteria which retain the primary stain. They are still violet. They do not lose it in presence of decolorizer. But there are certain bacteria, you can see here, they are susceptible. They are very susceptible to decolorizer. As you put decolorizer to them, decolorizer will take the primary stain out and they again will become transparent. So, in the third step, some bacteria are still violet, but some become transparent. That makes a difference, right? That makes a difference. You can see here. And lastly, when you provide the secondary stain, which is a red stain, so the bacteria which retain the primary stain, there is no space inside them. So they will not take the secondary stain because they are already saturated. They are filled. They don't have space, right? So these are still violet and these are known as gram-positive bacteria. So in the end, they are gram-positive. And the bacteria which were susceptible to decolorizer, they lose the primary stain. Now they are empty from inside. You can see they are empty from inside. So when you provide the secondary stain, which is a red stain, they will take the red stain and in the end they are red. The red one are the gram negative. So what is the summary? The violet one are gram positive, the red one are gram negative. Why the violet one are gram positive means why? What is the main step which differentiate them? It's decolorization. Now you may be thinking why some bacteria are resistant to decolorizer and why some bacteria are susceptible to decolorizer. You may be thinking that it's the difference is due to the peptidoglycan content, right? Now the cell wall, all bacteria have cell wall. There are some bacteria which have more peptidoglycan content in the cell wall. So here when you add the decolorizer, decolorizer cannot take the primary stain out because the peptidoglycan content is very thick and decolorizer cannot penetrate and take the primary stain out. But there are some bacteria in which the cell wall is there, but peptidoglycan content is less. So the decolorizer can easily penetrate inside the bacteria and take the primary stain out. That makes the difference. So basically here the difference is due to the cell wall composition. See the composition of the cell wall. In the cell wall, see the peptidoglycan content. Can you see the two types of bacteria shown to you here in this diagram? In this diagram, can you see this bacteria having a very little peptidoglycan content? It's gram negative. Peptidoglycan content is very less, you can see. And here you can see the peptidoglycan content is much more. It is thick peptidoglycan content. Can you see here? It's more. So this bacteria is gram positive. So when you give decolorizer here, they will not lose the primary stain. But when you give the decolorizer here, decolorizer can easily penetrate inside and they will lose the primary stain. That's why take the secondary stain. So you got the principle, I guess. So it's the integrity of the cell wall which decide whether the bacteria is gram positive or gram negative. It's the most important principle. Who will decide the bacteria is gram positive or gram negative? Who will decide it? It's the cell wall, the integrity of the cell wall, the peptidoglycan content of the cell wall. That will decide whether the bacteria is gram positive or whether the bacteria is gram negative. You got my point? Now, if I say there is a bacteria in which the cell wall is damaged. Imagine a gram positive bacteria having a thick peptidoglycan content. If the cell wall is damaged, can I say the gram positive bacteria convert into gram negative? Yes, the gram positive bacteria can be converted into gram negative bacteria if you damage the cell wall. It's the cell wall integrity which decide. And iodine is a mordant, I already told you. What do you mean by mordant? Mordant is something which form a complex with the primary stain. So it retains the primary stain inside the bacterial cell only. Primary stain is not leaking out, right? So that is the meaning of the mordant. Now based on gram stain, we have two type of bacteria. The violet one are gram positive and the red one are gram negative. So based on gram staining, we have two types of bacteria, the violet, the red, the gram positive, the gram negative. Based on shape also we have two types of bacteria, based on shape. The bacteria which are spherical in shape. Can you see? Spherical, circular in shape. These are known as cocci. And the bacteria which are rod shaped, elongated, rod shaped, these are known as bacilli. Got my point? So based on gram staining, the two types of bacteria, this is based on gram staining. And based on shape, again two type of bacteria, the cocci and the bacilli. Let's combine the two classification together. Let's combine the two. So we will be having four types of bacteria. The gram positive cocci, the gram negative cocci, the gram positive bacilli and the gram negative bacilli. So let's divide bacteria under four categories. This classification is based on combination of two things. The gram staining, number one, and the shape of bacteria, number two. So gram staining and shape of bacteria, based on these two things, we divide the bacteria into four categories. What are the four categories here? Can you please tell me? It's gram positive cocci, it's known as GPC, gram positive cocci. It's gram negative cocci, it's GNC, gram negative cocci. After that, gram positive bacilli, it's GPB, gram positive bacilli. And it's gram negative bacilli, it's GNB, gram negative bacilli. You got my point? So these are the four categories under which we divide the bacteria. Now, can you see the gram positive cocci? Since they are cocci, they are spherical, and since they are gram positive, they are violet. So you can see the shape and the color. They are spherical, they are violet. The gram negative one are the red, but they are cocci since they are spherical. So see the shape, see the color. So the purple spheres and the red spheres, they both are cocci, but one is gram positive, one is gram negative. In the same way, the purple rod and the red rod, you can see these both are rods. So both of them are bacilli, but the purple one is the gram positive and the red one is the gram negative. Now, based on the color and shape, size, it is the biggest clue given to you in the question. In the question, sometimes you will get the image-based question in microbiology in your exam. So in the image, they can show you the bacteria. You have to see the two things in the bacteria to decide its gram classification. So you have to see its color, whether it's violet or red, and you have to see its shape, whether it is spherical or rod shape. Based on these two things, you can decide the category. Now you have to learn the examples under each category. You have to learn the examples. What are the examples here? Gram-positive cocci are only two. S. Staphylococcus, Streptococcus. That's it. The 2 gram-positive cocci. Staphylococcus, Streptococcus. That's it. Again, the 2 gram-negative cocci. We have only 2 gram-negative cocci. The Neisseria and Moraxella. The Neisseria, the Moraxella. So we have 2 gram-negative cocci. The Neisseria, the Moraxella. After that, gram-positive bacilli. We have 5. So there is a mnemonic for you. The mnemonic is A, B, C, D, L. A, B, C, D, L. A stands for actinomycetes. B stands for bacillus. Bacillus anthrax, all bacillus. C stands for clostridium. Various types of clostridium, all of them. And D stands for diphtheria. You can say corynebacterium diphtheria or diphtheria. And L is Listeria monocytogenes. So these are the 5 bacteria you have to learn. A, B, C, D, L. These are gram-positive rods, gram-positive bacilli. And apart from these all, this is A, B, C, D, L. A, B, C, D, L. Apart from this 2, and this 5. Remaining all bacteria fall under the 4th category. That is gram-negative cocci. That is the 4th category. Now don't learn the list here. It can be E. coli, it can be Salmonella, Shigella, Vibrio, Klebsiella, Proteus. It can be rickets, spirochetes. It can be any other bacteria. So all other bacteria except these fall in the 4th category. Now this is an ultra important classification. You cannot afford missing it. This is the basics of microbiology. If you don't know this classification, you cannot solve any clinical based question based on bacteriology. If you get any clinical based question, clinical scenario based question in microbiology, in bacteriology especially, you will get a clue in the question. The bacteria is gram-positive cocci, gram-negative cocci, gram-positive bacilli, gram. So you should have a differentials in your mind. If the examiner is saying there is a patient having such symptoms, we have taken the specimen and we have observed it is a gram-positive bacilli. So the 5 options come in front of you, A, B, C, D, L, right? Among 5, 1 is the option. Right. So you can see other properties, other things, other clues given to you, and you can rule out certain options. So you should know this classification very well. That's all about gram staining. Right. Now after this, let's start acid fast stain. Acid fast stain is also known as Ziehl-Neelsen stain. Why it is known as Ziehl-Neelsen stain? This stain is not discovered, but it is modified by Ziehl and Neelsen. It is discovered by a scientist known as Erich, but it is modified later on by Ziehl and Neelsen. That's why known as Ziehl-Neelsen stain. Now here also we are having 4 steps, just as same as that of gram stain. You can compare. The steps are same, but the components are different. Now here also we take a primary stain. Here the primary stain is a red stain, not the violet like gram stain. So here any red stain will work. Usually we use carbol fustian, right? Here the mordant is heat, not iodine in comparison to gram stain, right? The compare and contrast the both. So what is mordant? Mordant is something that fixes the primary stain inside the bacteria. So here mordant is heat, right? The third is the decolorizer. Here we use decolorizer acid. It's sulfuric acid. It's H2SO4. Now here, apart from the acid, you have to learn its concentration. It's 20% standard we use. So standard we use 20% sulfuric acid, 20% H2SO4 as a decolorizer in acid fast stain. In gram stain, if you remember, it's acetone or alcohol. So here it's acid. And the last is the counter stain. Counter stain is the secondary stain. It's counter stain. It's any blue stain. It's any blue. So you can see the primary and secondary stains are reversed here in contrast to the gram stain. In gram stain, the primary stain was violet and the secondary was red. But in acid fast, the primary is red and secondary is not violet but it's blue. You got my point? So at the end, you have 2 types of bacteria. The bacteria which retain the primary stain. They are red in color and these are known as acid fast organism. Acid fast organism. Or else you have the bacteria which retain the secondary stain or counter stain. That is, they are blue in color, and these are known as non-acid fast organism. So some bacteria are retaining the primary stain and some bacteria are retaining the secondary stain. So some are red, some are blue. That is, some are acid fast organism, some are non-acid fast organism. You may be thinking why there is a difference. Why there is a difference in the two type of bacteria? Why some are retaining primary stain and why some are retaining the secondary stain? To understand this, you have to see this diagram. Can you see? You know the basic principle. All bacteria are transparent. By nature, they all are transparent. So here you can see a transparent bacteria. Here also you can see a transparent bacteria. Right? Now you give a primary stain which is red in color. You give a primary stain which is red in color. So this also become red and this also become red since they were transparent. Now you give the mordant as heat. The mordant is the heat. So heat is fixing the primary stain. After that you give the decolorizer. The most important step, you give the decolorizer which is 20% sulfuric acid. So there are some bacteria which retain the primary stain, like they are resistant to the acid. They are not getting decolorized by the acid, right? That's why they are retaining the primary stain. That's why they are still red. They are still red in color. But on the contrary, there are some bacteria which are susceptible to the acid. They are susceptible to the decolorizer. So they lose the primary stain and they again become transparent like the beginning. You got my point? Now you add a secondary stain which is a blue stain. Any blue stain like methylene blue, right? So the bacteria which resist the decolorizer, they don't have space inside them. So they will not entertain, they will not allow the secondary stain to enter them. So at the end, they are still red in color. And these are known as acid fast organism, right? And the bacteria which are susceptible to the acid, so they become transparent in the last step. Now if you add the secondary stain, they take the secondary stain and in the end they will become blue in color. And these are known as non-acid fast organism, right? Now the point is that why some bacteria are resistant and some bacteria are susceptible to the acid? It's because of mycolic acid content in their cell wall. The bacteria which have more mycolic acid content or mycolic acid content present in their cell wall, they do not get decolorized. They do not allow H2SO4 to go inside and take out the primary stain. But the bacteria which do not have mycolic acid, so acid will do the decolorization in them. So the reason here is mycolic acid. If you compare it, the principle with gram stain, in gram stain it was peptidoglycan content of the cell wall, that makes the difference. And here it's mycolic acid content of the cell wall, that makes the difference. So this is how you can learn the differences. So this is acid fast and non-acid fast organism. What do you mean by the term fastness? It's not related to the speed, the slow, the fast. No, it's not like that, right? What do you mean by fastness? The word fastness here means resistance. It's resistance, right? Acid fast organism, don't read like that. Read acid resistant organism. Yes, they are resistant to H2SO4. That's why, because they are resistant, na? They are not getting decolorized by acid. That's why known as acid resistant organism, acid fast organism. And non-acid fast, read fast as resistant. So these are non-acid resistant organism. Non-acid resistance means they are susceptible to H2SO4. So basically the meaning of fastness, it's mean that resistance. So that is the meaning of the fastness. At the end, you can see the two images. Here you can see in both of them the red bacilli. You can see the red color bacilli in a blue necrotic background. So you can see the red, both of them are acid fast organism. Actually, it is an image for Mycobacterium tuberculosis. You can see it is an acid fast organism. That's why the organism is red in color, but the background is blue. Can you see that? Can you see? So you can see the principle of the gram stain as well as acid fast stain here. So can you see the first diagram? In the first diagram, appreciate the peptidoglycan content in the cell wall. I can see a thick peptidoglycan content in the cell wall, right? That's why it's gram positive. In the second diagram, I can see a thin layer of peptidoglycan content in the cell wall, a thin layer. That's why it's gram negative. So peptidoglycan more is gram positive, peptidoglycan less is gram negative. And the third one, mycolic acid present in the cell wall. If mycolic acid is present in the cell wall, it's acid fast organism. So you can clearly see the differences, the principle of gram stain and acid fast stain here. It's cell wall. But which component of cell wall, that makes a sense, that makes the difference. So coming on acid fast organism, how many acid fast organisms do you know? So we are dividing the organisms into two categories, the acid fast and non-acid fast. These all are acid fast organisms. This is the list given to you. And except this list, all other organisms fall in non-acid fast. So don't learn non-acid fast. Learn this list and remaining all are non-acid fast. So for learning this list, I am having a mnemonic for you. Again, I am having a mnemonic for you. Now listen, microbiology is full of mnemonics. I will give you in the entire syllabus, I will give you multiple mnemonics to make your learning more easy, to make your retention more powerful. Mnemonics will help you in retaining the list, the big list, right? So there are two types of knowledge, the factual and the conceptual. Wherever there is a conceptual thing, now I will explain you the concept. You don't have to mug it up. But there is a factual knowledge. The factual you have to mug up. There is no... Now this is a list. I cannot explain the list now. You have to mug it up. This is a list. But for that I will provide you mnemonics to make it easy, right? So the mnemonic is no longer separate booking for RIM, R-I-M, RIM, SIM card, right? Let me tell you the full form. N stands for nocardia. L stands for Legionella. S as it stands for Smegma bacilli, right? B stands for bacterial spores, right? R-I-M. R stands for Rhodococcus. I stands for Isospora. M stands for Mycobacterium, right? S stands for sperm, the male sperm, human male sperm. And C stands for Cryptococcus and Cyclospora. So you have to try to say the full form again. What is the full form? It's no longer separate booking RIM, R-I-M, SIM card. So say the mnemonic first. No longer separate booking RIM, SIM card. Now try to see the full form. It's easy. So N stands for nocardia. L stands for Legionella. S stands for Smegma bacilli. B stands for bacterial spores. R stands for Rhodococcus. I for Isospora. M for Mycobacterium, both Mycobacterium, tuberculosis as well as Lipri. S stands for sperm. There are two S, one is spore, one is sperm. You have to understand. One is spore, the bacterial spore, right? Smegma bacilli, and another S is sperm. And B say hai bacteria, but it's bacterial spores. And C stands for Cryptococcus and Cyclospora. So out of them, this Cryptococcus is the fungus, right? And this Cyclospora and Isospora are the parasite, and remaining all are bacteria, except human sperm. Remaining all are bacteria. So maximum are bacteria. There is one fungus and two parasites also. So this is a complete list of all organisms, or I must say not organism, the structures, because human sperm is also included now. So all the structures which are acid fast. Learn the mnemonic. With mnemonic it will be easy, right? Now there are some modifications in acid fasting. Let me tell you what are the modifications in acid fasting. So standard we use 20% H2SO4. I told you, standard we use 20% H2SO4. But we can change the percentage according to the organism. For Mycobacterium Lipri, not for Mycobacterium tuberculosis, mind my words, right? For Mycobacterium Lipri, instead of 20%, we use 5% H2SO4. It's a very important MCQ. Please learn, right? For Nocardia, we have to use 0.5% H2SO4. Right. For oocyst, for Cryptospora, Isospora and bacterial spore, we use 0.25% of H2SO4. So 5, 0.5 and 0.25, not 20, and standard is 20. Right. So there are two types of Mycobacterium. Mycobacterium tuberculosis and Mycobacterium lepra. I told you for Mycobacterium lepra we use 5%. Right. We use 5% H2SO4, just now I told you. For tuberculosis we use standard 20%. Right, you can see, right? So can I say Mycobacterium lepri is less acid fast as compared to Mycobacterium tuberculosis? Because it get decolorized only at 5% H2SO4, but Mycobacterium tuberculosis required 20% H2SO4. Yes. So we can say this line very easily that Mycobacterium lepri is less acid fast than Mycobacterium tuberculosis. Now don't mug up this line. Please understand the concept behind that. Right. So you know the two type of percentage of the H2SO4 required to decolorize the two type of the bacteria. So that was the concept behind that. Now let's compare and contrast the gram stain and acid fast stain. Please do the comparison and contrasting here. So what is the primary stain in gram stain and what is the primary stain in acid fast stain? In gram stain, the primary stain is any violet stain, like gentian violet, crystal violet, and in acid fast stain, the primary stain is any red stain like carbol fission. The next step is the mordant. What is the mordant in gram stain? It's iodine. And what is the mordant in acid-fast stain? It's heating. What do you mean by mordant? It is something which is fixing the dye inside the bacteria. Right. Now coming on the decolorizer. The decolorizer, the most important step. Can you tell me the decolorizer in gram stain? It's acetone or alcohol like ethanol. And what about the acid-fast stain? It's an acid. Here acetone or alcohol but not acid. And here we use the acid. It's 20% sulfuric acid. And what about the last step? Counter stain. Counter stain. So counter stain is the secondary stain. In gram stain, it's any red stain like carbol fustian. But in acid-fast stain, it's blue stain like methylene blue. Now see the primary and the secondary stain. You have to compare and contrast each step. Now at the end in gram stain, some are violet which retain the primary stain. So these are gram positive, and some are red which are known as gram negative. See the color. Violet and red. Violet are gram positive and the red are gram negative. And in acid-fast, at the end, some retain primary which are red in color. These are known as acid-fast organism, and some take the secondary stain which are blue in color, that is a non-acid-fast organism. So you can see violet and red are the end result of gram stain, and blue and red are the end results of acid-fast stain, right? So you got the concept behind them. Now let's come on the third stain, the third type of differential stain, the Albert stain. In differential stain, we are having three types of differential stain, the gram stain, the acid-fast stain, the third one is the Albert. So let's talk about Albert stain. So Albert stain differentiates all the bacteria which have metachromatic granules from the other bacteria which do not have metachromatic granules. Right. So there are some structures inside bacteria known as metachromatic granules. Some bacteria have it, some do not have it. So the bacteria, there are 5 bacteria, I will tell you the list, having metachromatic granules. All the 5 bacteria can be stained by Albert stain. Albert stain is a stain which is used to differentiate the bacteria based on metachromatic granules, you can say. So let's start Albert stain. Now one of the bacteria which is having metachromatic granules is Corynebacterium diphtheri. The Corynebacterium diphtheri is one of the bacteria having metachromatic granules. So when we apply Albert stain on Corynebacterium diphtheri, you can see the bacilli are green color. Can you see the bacilli? It's green color. It's green color bacilli. So all of them are green color bacilli, number 1. So green color bacilli, and at the poles, both poles, there are blue color granules. The blue color metachromatic granules. Here in the diagram also you can see. At both the ends, the blue granules are present. So the bacilli is green color and the granules are at both the poles, they are bluish black metachromatic granules. It's in Corynebacterium diphtheri. On Albert stain, the diagram is like this. Now the next important point to be understood here, you can see most of the bacteria are either L shape or V shape. Can you make it out? L or V. The bacteria are either L or V. It is looking like some Chinese language is written. So that's why it is known as Chinese letter arrangement, or also known as cuneiform arrangement. Ultra important MCQ and ultra important image based question also, a typical spotter image. So if this image is coming in your exam with a green bacilli, green color bacilli, the structures of the V or L, and at the poles there are blue color granules. It's typical diagram of Corynebacterium diphtheri on Albert stain. It's not gram stain, it's not acid-fast stain, it's the Albert stain. So learn the colors also. The bacteria is green color, the metachromatic granules is blue color, and they are V-shaped or L-shaped. Now can you tell me the reason why they are V or L-shaped? This is a bacteria. When a bacteria divide, the 2 daughter bacilli are formed. This is a daughter bacilli and this is a daughter bacilli. So basically they got separated normally. But here in Corynebacterium diphtheri, it's the incomplete separation of the daughter bacilli. So both of these daughter bacilli are not completely separating from each other. That's why known as V-shaped or L-shaped. You got my point? So that is the V or L shape. Now what are the steps for the Albert stain? First, you heat fix the smear. You make a smear, you heat fix it. Then you first place Albert 1 stain for the 5 minutes and without washing. Don't wash it in between. Just drain the solution and then put Albert 2 for another 1 minute. And in the end wash it. And see under microscope you will get this diagram. So what is Albert 1 and Albert 2? Albert 1 is toluidine blue and malachite green. It is toluidine blue, malachite green, glacial acetic acid, alcohol and water. But Albert 2 is iodine in the form of the potassium iodide. You should know the composition of Albert 1 and Albert 2 for MCQ purpose. You should know. Now, what are these granules based on which we divide the bacteria into two categories? The bacteria which have these granules, they are stained by Albert stain. But the bacteria which do not have these granules, they are not taking the Albert stain, right? So, these granules, we can see the granules at the end of the bacteria. No? I'm drawing Corynebacterium diphtheria. I can see the granules at the poles. Since the granules are present at the poles, let me draw a bacteria. And the granules are present at the pole. By pole, I mean the north pole, the south pole, at the two poles. These are known as polar bodies. So, metachromatic granules are also known as polar bodies, number one. Number two, they are known as metachromatic because they are blue in color. I am drawing it with blue color. That's my blue or violet color. So, it is known as metachromatic, reddish violet, bluish violet. Polar bodies because they are present at the pole of the bacteria. Now, name the scientist who discovered the polar granules. The name of the scientist is Babes Ernst. Babes Ernst. That's why known as Babes Ernst granules. Babes Ernst granules. And there are 5 bacteria in which these granules are present. One of them is volutans, Spirulum volutans. So, in the first bacteria it was discovered, it was volutans. It is also known as volutin granules. So, basically these all are synonyms. Whether I say volutin granule because it is the name of the bacteria in which it is discovered, or I say Babes Ernst granules, or I say polar bodies, or I say metachromatic granules. I can say anything of it. These are the 4 different names given to the same metachromatic granules, right? Now, what is the composition? What they are containing inside them? The composition is the phosphate. They contain phosphate inside them. They contain phosphate inside them. The composition is the phosphate. Now, why phosphate is present? Normally, when the bacteria go on dividing, the phosphate is utilized in forming the nucleic acid of the daughter cell. But during the starvation, the bacteria do not divide. If the bacteria is not dividing, the phosphate is not utilized. So, it will accumulate at the end of the bacteria. So, basically, the metachromatic granules indicates the bacteria is not dividing, it is in starvation state. So, that is about the metachromatic granules. Now, can you tell me the name of the 5 bacteria in which these metachromatic granules are present? And can you tell me the name of the 4 stains by which we can stain the metachromatic granules? Can you tell me the 5 bacteria and the 4 stains? The 5 bacteria and the 4 stains for metachromatic granules. Now, you can get a MCQ either on the bacteria or you can get a MCQ on the stains. That is all about metachromatic granules. So, for bacteria, I am having a mnemonic. Have you visited Agra? Anytime. There is a garden in Agra. The name of that garden is MHC. No, it's not there. Just for making mnemonic, I have created this mnemonic. It's MHC Garden Agra. So, let me tell you the full form. M is Mycobacterium. Mycobacterium, all Mycobacterium are there. S is Spirulum volutans, the first bacteria in which it is discovered, that's why known as volutin granules. The C is Corynebacterium diphtheria. C is known as Corynebacterium diphtheria. Garden is Gardnerella vaginalis. Garden is Gardnerella vaginalis. And Agra is Agrobacterium tumefaciens. So, these are the 5 bacteria in which these granules are present. Now, can you tell me the stains by which we can stain them? I know one of them, that is Albert, which currently I am teaching you, Albert stain. But apart from Albert, we can stain with many other stains also. So, the mnemonic is PLAN, P-L-A-N, PLAN. So, P stands for Ponder stain. L stands for Loeffler's methylene blue. It is the most common stain which is used for staining these granules. A is Albert. Currently, I am teaching you Albert. On Albert, the bacteria is green, but the granules are bluish in color. And N stands for Nizar. So, please learn the full form of MHC Garden Agra and please learn the full form of PLAN. So, MHC Garden Agra are the bacteria, the 5 bacteria inside which these granules are present. And PLAN, P-L-A-N, PLAN are the 4 stains by which we can stain these granules, right? So, that is all about metachromatic granules. Now, in the end, I would like to add a note on a special stain which is known as bipolar stain. What is bipolar staining? Have you seen a safety pin? Now, normally, okay, let me tell you. This is a bacteria. Imagine this is a bacteria. Now, when you stain this bacteria, I'm talking about any stain. It can be Gram stain, acid fast stain, Albert stain, simple stain, any stain. So, usually the stain distributes uniformly throughout the bacteria. It is normal staining pattern, whatever it is distributing uniformly. But there are certain bacteria which accumulate the stains at the end, which accumulate the stains at the poles. There are certain bacteria which accumulate the stains at the ends or the poles. So, it is known as bipolar staining. Can you see the word bipolar? Bipolar, because the stain is present at the two poles. That's why known as bipolar staining. Now see this diagram and imagine a safety pin. Have you seen a safety pin at your home? You may have seen it. It is a multi-purpose pin, right? So this is a safety pin you can see here. So you can see it's looking like the stain at the two corners, at the two poles. So it's known as safety pin appearance. It's a very important MCQ. It is known as safety pin. So in this diagram also I have shown you multiple bacilli. If you can see all the bacilli, you can appreciate the stains are present at the poles. The stains are present at the poles of the bacteria. Can you see the stains are present at the poles? Here also the same thing is shown to you. You can see the stains are present at the poles of the bacteria. Yes. So that is the bipolar staining or safety pin appearance. Now can you tell me the name of the bacteria in which there is no uniform staining, the staining at the poles? There are 5 bacteria. Again you have to learn the list. I told you now microbiology is full of list and mnemonics. Wherever there is a list I will provide you a mnemonic. So the mnemonic is can't produce very high yield. You can learn the mnemonic like this. This is a safety pin. Let me draw a rough diagram of a safety pin. Safety pin cannot produce very high yield. Learn the mnemonic like that. Safety pin cannot produce very high yield. So say the full form. C stands for Campylobacter. P stands for Pseudomonas malae or Pseudomalae. V stands for Vibrio, but please mind it, Vibrio cholerae, it's not Vibrio cholerae, it's Vibrio parahemolyticus. High ka H stands for Haemophilus ducrei. Please mind it, it's Haemophilus ducrei, it's not Haemophilus influenzae. And yield Y, it's Yersinia. So can't produce very high yield. What is the mnemonic? Can't produce very high yield. Can't produce very high yield. So it's Campylobacter, it's Pseudomonas malae, it's Vibrio parahemolyticus, it's Haemophilus ducrei and Yersinia. Please say the full form. So that's now after all these there are some special staining methods for special organelles. So for spores of the bacteria, we can use either acid-fast stain. We have seen what was the mnemonic of acid-fast stain? No longer separate booking for REM-SEM card. No longer separate booking. What was B for booking? It's bacterial spores, if you remember. Now correlate the things. Try to correlate it. So in acid-fast stain, we use 0.25% of the sulfuric acid. It was coming in the modifications also. We don't use 20%. We use only 0.25%. Malachite green. For spores, we can use malachite green also. And for spores, we can use a phase contrast microscope. One of the type of the microscope. A phase contrast microscope. It is the best method. It is the best. I must say it is the best method. Right. For lipids, we use Sudan black. It's a very important MCQ. I guess you may be knowing that. For lipid or for adipose tissue, for the fat lobules, we use Sudan black. There are many types of Sudan blacks available. They are used for the lipids. For carbohydrates, especially for glycogen, we use iodine stain. For the flagella, we use tannic acid staining. It is known as Lefson method. Now, these all are your MCQs. Now, don't go in the details of all these. Just learn the names. That is more than sufficient. So, we have completed the various types of staining methods. Let's revise it quickly. What we have learned in this session. Can you help me in the revision? Now, we have learned the 2 types of preparations, the unstained preparations and the various types of stained preparation. Now you have to help me. So unstained preparation are known as wet preparation because we have to see them in the wet state only. These are also known as hanging drop preparation. So these are basically used to see the motility of the bacteria. These are not used to see the morphology. These are used to see the motility, right? Now stain preparation, we have various types of staining available. The simple stain, it gives same color to all bacteria. The examples are methylene blue and basic fustian. The negative stain, it stains the background, not the structure to be demonstrated. It stains the background, not the bacteria. The examples are India ink and nigrosin. The examples are India ink and nigrosin. The third is the impregnation method. Impregnation means layering. It is used for 2 thin structures like the bacterial flagella, like spirochetes, right? Here, it means layering. So, we do the layering with silver. The example is silver. Now, coming on differential stain. The differential stain, it provides different color to different bacteria, right? Here, gram stain. Gram stain, you already know the 4 steps. You know the principle. The principle is the peptidoglycan content of the cell wall. The bacteria which is having more peptidoglycan content of the cell wall, they are gram positive and they are violet in color. They retain the primary stain. And the bacteria which is having less peptidoglycan content in the cell wall, they are gram negative. They are red in color and they take the secondary stain. They lose the primary stain, they get decolorized by the decolorizer. And you know the 4 steps, right? That is gram staining. The acid-fast staining, you again know the 4 steps of the acid-fast staining. At the end, we have acid-fast organism and non-acid-fast organism. You will be knowing the list for that. And Albert stain differentiate the bacteria having metachromatic granules from the bacteria who do not have metachromatic granules. So, there are 5 bacteria which are stained by Albert stain. One of them is most important, Corynebacterium diphtheria. But you know the mnemonic, it's MHC Garden Agra. The 5 bacteria which are stained by Albert stain. So, this stain differentiate this 5 bacteria from all others. What are metachromatic granules? They are phosphate accumulation. Why they are present in the bacteria? Because in starvation, bacteria is not dividing. You may be knowing that, right? And name the stains for metachromatic granules. The 4 stains are PLAN, PLAN. You know the full form. Now, the last we have learned here is the bipolar staining. Bipolar staining is the stain which accumulates at the poles of the bacteria. At the poles of the bacteria, the stain is accumulating. It is not distributing uniformly in the bacteria, giving an appearance of the safety pin. That's why known as safety pin appearance. So, how many bacteria have such appearance? There is a mnemonic can't produce very high yield. You know the full form. It's Campylobacter, it's Pseudomonas malae pseudomolai, it's Vibrio parahemolyticus, it's Haemophilus ducrei, not influenzae, and Yersinia. Right? So, these are the various types of the stains we have learned, and we have compared and contrast the gram stain and acid-fast stain. You have to compare the primary stain in both of them. You have to compare the mordant in both of them, compare the decolorizer in both of them, and compare the secondary stain in both of them. And see at the end what colors we are having, the gram positive, gram negative, the violet and the red color, and acid-fast organism and non-acid-fast organism, the red and the blue color. So, here we have violet and red, here we have red and blue. You have to understand and you have to compare and contrast each step here. That is about that. Based on gram stain and acid-fast stain, based on gram stain, we divide the organisms into 4 categories. You can see the 4 categories here. We have seen gram positive cocci, gram negative cocci, gram positive bacilli, gram negative bacilli. You can see the bacteria inside that. I have provided you mnemonics also here. The gram positive cocci are Staphylococcus and Streptococcus. The gram negative cocci are Neisseria and Moraxella. The gram positive bacilli is A, B, C, D, L. You know the full form. It's Actinomycetes, Bacillus, Clostridium, Diphtheria and Listeria. And remaining all of them are gram negative bacilli. You got my point? So, that is about the gram staining. We divide the organism under 4 categories. But what about acid-fast staining? In acid-fast staining, we have only 2 categories. The acid-fast organism and non-acid-fast organism. So, out of the 2 categories, you have to learn the list of acid-fast organism. Remaining all will be non-acid-fast. So, in acid-fast, we have seen the mnemonic. The mnemonic is no longer separate booking for RIM, R-I-M, RIM. SIM card, no longer separate booking for RIM SIM card. It's a mnemonic, you know. Now, try to see the full form. Again and again, we have discussed the full form, right? So, let's solve some previous year questions based on this concept. We have done the summary also. So, let's solve some PYQs based on the staining techniques. So, let me discuss the question. So, the first question is in front of you, the first PYQ. Let me read it. One of the following staining method is an example of negative stain. Negative stain. Read the 4 options. Is it gram stain? Is it Fontana stain? Is it India ink preparation? Or is it Ziehl-Neelsen stain? What is negative stain? Negative stain is something which stain the background but not the structure to be demonstrated, not the bacteria. So, what are the examples? I told you, yes, the correct answer is India ink. I told you in this session the 2 examples of the negative stain. It's India ink and Negrosin. So, the correct answer among them is of course India ink. You can see the image also of the negative stain. The correct answer is India ink. India ink and Negrosin are the 2 examples of negative stain. The second question is in front of you. In negative staining, what is correct about it? Can you read the 4 options please? So, the structure to be demonstrated is stained. Is it true or false? No, it's false. The structure to be demonstrated is not stained. No, this statement is false, right? The background is not stained. Is it true or false? No, it's also false because here the background is stained. Background is not stained, it's false statement. Now, the next is background and substance both are stained. No, Only background is stained, but the structure to be demonstrated is not stained. So this statement is also wrong. But read this statement. The structure to be demonstrated is not stained. Yes, I guess this statement is correct. The structure which you want to see, like the bacteria, you don't stain that. Stain the background. So the correct answer here is B. It's a little bit tricky question, but you can make it out if you know the concept. So the correct answer here is B. The structure to be demonstrated is not stained. See, in this image, you can see the structure, that is the bacteria, the capsulated bacteria diplococci. It has to be demonstrated, it is not stained, it's still transparent. We are staining the background so that transparent bacteria can stand out in a dark negative background. So that is the thing. Coming on the next question, let me read the next question in front of you. Can you arrange the order of the gram staining from the 4 options given below? So, mordant, acetone or alcohol, crystal violet and diluted carbol fission. Now you know the steps of the gram stain. I have described the steps of the gram stain. Can you arrange them in a order? Can you arrange them in a order from first to last? Now let me revise you the 4 steps of the gram staining. So first we add a primary stain, then we add mordant, then we add decolorizer, and then we add secondary stain. So primary stain we use is any violet stain like crystal violet, mordant is iodine, decolorizer is alcohol or acetone, and secondary stain is any red stain like carbol fission. Now see the order, it's in this order. We are using in this peculiar order. So crystal violet, that is 3, followed by mordant, that is 1, 3, 1, then alcohol or acetone, 3, 1, 2, and last, in the last there is diluted carbol fission. So 3, 1, 2, 4. 3, 1, 2, 4 is the correct sequencing, is the correct sequence here. Can you make it out? No, it's a tricky question. It's a tricky question. It's a question from INI-CET PYQ. It's a tricky question, but yeah, it's very simple. If someone knows the concept, you can make it out. So the correct answer here is this one. It's 3, 1, 2, 4. You have to know the 4 steps. If you know the 4 steps, you can arrange the sequence. We have discussed it. So coming on the next question. The next question is in front of you. Can you see? Gentian violet coloration of the gram-positive bacteria is due to. Why gram-positive bacteria are violet in the end? That I want to ask. Why gram-positive bacteria are violet in the end? What is the reason? Do they have peptidoglycan content in the cell wall that gives them violet color? Do they have a capsule that gives them a violet color? Do they have a cell membrane or none of the above? What you will say? Yes. So examiner want to ask you the principle of the gram staining. Yes, the correct answer is peptidoglycan content. The gram-positive bacteria, they have more peptidoglycan content in their cell wall, so they resist the decolorizer to enter and take out the primary stain. Right? So they retain the primary stain and resist the decolorization. So the correct answer here is the peptidoglycan content of the cell wall. Here in the diagram, you can see the purple color is the peptidoglycan content. Here a very less peptidoglycan content is shown, and here in this bacteria abundant of peptidoglycan content is shown. So this is gram negative, this is gram positive. So it's peptidoglycan content of the cell wall that makes the difference. Got my point? Now the next question is in front of you. Can you see the question? Let me read it. So a HIV patient, the patient is HIV. The first clue, pick up the clues from the question. The patient is HIV positive, presents with fever, chronic diarrhea. Diarrhea is another clue given to you. Fever and diarrhea. So patient is having some GIT infection, right? GIT infection. Presented to the OPD. Stool was examined. The stool was examined and stool shows oocyst. So can you tell me the causative organism of diarrhea in a HIV patient whose oocyst is present in the stool? Compile all the clues and try to come on the diagnosis. The diagram of the oocyst is given to you. The oocyst I can see. Now come on the step by step we will go ahead. I can see the oocysts are of red color. That is, can you identify the stain? The red oocyst on a blue background, on a blue necrotic background. Yes, the stain is acid-fast stain. It's an acid-fast stain. And the organism, the structure here is acid-fast organism. The acid-fast organism because they are red in color. We know acid-fast organism are red in color on a blue background. So can you tell me the causative agent of diarrhea in a HIV patient whose oocysts are present in the stool and they are acid-fast organism? Yes. You know the acid-fast organism, I have given you the mnemonic. No longer separate booking, rim, SIM card. If you try to say the full form now, it's isospora and cryptospora. So two spores are present. It can be isospora, it can be cryptospora, I cannot make it out. Both can be there. They can be there. It can be isospora, it can be cryptospora, because both of them cause diarrhea in HIV patient. Both of them. So among all these are the two causative organisms which can cause diarrhea in a HIV positive patient and whose oocyst can be present in the stool. So the answer is isospora or cryptospora. But what the examiner want to ask us? Examiner want to ask us the primary stain. So for the oocyst of isospora and cryptospora, all of them are diluted carbol fustian. Diluted carbol fustian or strong carbol fustian. So is it 20%? Is it 0.5%? Is it 1% or 5%? So I told you in the exceptions, isospora and cryptospora, normally we use 20% H2SO4, but isospora and cryptospora come in the modifications of the acid fast stain. Here we use 0.25 to 5% of the H2SO4. 0.25% is not given in the option, I will go with 5%. That's why I will go with 5%. So the correct answer here is B. Now the examiner have tested multiple knowledge in this one question. The first thing examiner tested whether you know that what is the cause of diarrhea in a HIV patient. The second thing he has tested, do you know the oocysts which are present in the acid fast organism? Can you identify the stain, the image? It's the acid fast stain showing the acid fast organism, the red color oocyst, right? And he has tested whether you know the exceptions, the modification of the standard acid fast staining. So he has tested multiple, it's a very difficult level question, but step by step, if you know the concept, you can make it out, right? Coming on the next question is in front of you. Can you read it? Decolorization in Zeden stain. So what is the standard decolorization? They are not giving any name, so I will go with standard. So in Zeden stain is Ziehl-Neelsen stain. That is acid fast stain. The four options are in front of you. Is it 1%, 5%, 10% or 20% sulfuric acid? So yes, if nothing is asked, I will go with 20%. But if the same question is asked for Mycobacterium lepra, my answer will become 5%. My answer will become 5%. So in this way, we can make it out. Here nothing is written, I will go with 20%. So standard is 20%. But there are some exceptions, I have mentioned that. So the next question is in front of you. Albert stain is used for. So Albert stain is a stain which differentiate the bacteria having metachromatic granules from the bacteria which do not have metachromatic granules. There are 5 bacteria which have metachromatic granules. Albert stain all of them. So what is the mnemonic I have provided you? It's MHC Garden Agra. So can you tell me the full form please? M stands for Mycobacterium, which is not there in the mnemonic. S stands for Spirulum volutan, which is not given in the 4 options. C for Corynebacterium diphtheria, which is one of the option. And Gardnerella and Agrobacterium is also not given in the options. So among the 5, only Corynebacterium is given here. So I will go with Corynebacterium, right? So the correct answer here is Corynebacterium diphtheria. You can see on Albert stain, the bacteria are of green color. In the Chinese letter arrangement, cuneiform arrangement, and at the poles they have the granules. So the correct answer here is B. Coming on the next question. The next question is metachromatic granules are stained by. I told you the name of the 4 stains. Which stain metachromatic granules? The mnemonic is PLAN. P-L-A-N, PLAN. Can you tell me the full form please? The Ponder stain. Yes, the correct answer is Ponders only. Yes. L, is it for Leishman? No. L is for Loeffler's methylene blue. It's not Leishman, the most common stain for metachromatic granules. It's Loeffler's methylene blue, it's not Leishman. A for Albert, it is not given in the option. And N for not negative stain. N is Neither. It's not there in the options. So don't just learn the mnemonic, you know. If you learn the mnemonic, you will get confused. P, A and N. P, L and everything is coming. But N is not negative stain, it's Neither stain. L is not Leishman, it's Loeffler's methylene blue, right? The correct answer is Ponder. Go with Ponder. So the mnemonic you should know and the full form of that plan you should know. So here P is for Ponder. Coming on the next question, it's in front of you. Modified Ziehl-Neelsen stain is used for. What is the modification? We do the modification in the percentage of H2SO4. Instead of standard 20%, we use some other percent like 5%, 0.5% and 0.25%. Do we do it for Mycobacterium tuberculosis? No, we don't do that. Do we do for Mycobacterium bovis? No. We do it for Mycobacterium lepri. Instead of 20%, we use 5%, but it is not there in the option. The next is the nocardia. Yes, we can do it for the nocardia. So exactly for nocardia, we use 0.5%. That is a modification, right? So all the organisms showing modification, among them only nocardia is given in the options. So I will go with nocardia. Going to the next question. So bacteria with safety pin appearance. What is safety pin appearance? So safety pin appearance is the bacteria in which the stains accumulate at the poles. That is known as safety pin appearance, also known as bipolar staining. So I had given you a mnemonic, can't produce very high yield. Can you tell me which among them is coming in the options? So can you see? H is not H influenzae, it's Haemophilus ducrei. I told you it's Haemophilus ducrei, not influenzae. V is, here V is coming now. This V is Vibrio parahemolyticus, which is our answer. Vibrio vulnificius is not there and Salmonella is not there. So among the full form, if you see in the mnemonic, it's Vibrio parahemolyticus which is coming here. So correct answer here is B. Can't produce very high yield. V is for Vibrio parahemolyticus, which is our correct answer here. H is there, but Haemophilus ducrei, not influenzae. So don't get confused, you should know the full form correctly, right? Now coming on the next question. All of the following organisms show bipolar staining except. The question contain except. So again apply the same mnemonic, can't produce very high yield. Let me apply the mnemonic. I guess Campylobacter granulomatans C is coming. I guess Yersinia pestis, it's Y coming. I guess Pseudomonas malai P, it's there. But H is not influenzae. Again and again I am highlighting here H is there. But it's H. ducrei, Haemophilus ducrei, it's not influenzae. So incorrect among them is D, and in the question except is asked. So I will go with D. So please learn the full form. The mnemonic is not important, but the full form is important. So we have covered a very important, ultra super duper important portion in the microbiology, that is staining techniques. You can secure many MCQs in your exam if you have the staining techniques, all the concepts behind it, if you know, and if you know all the list. The question can be direct or the question can be in the form of a clinical, clinical scenario based question. There will be clues based on the staining techniques, right? So we have finished that. Now before ending the session, let me tell you something. So sometimes now what happens, now you have to study for long hours, for many years. You will complete your MBBS. After MBBS, you will do your internship. You will prepare for your competitive exams for the post-graduation. You will enter in post-graduation. You will do your post-graduation. After that, you will do, masters. You will do a super master degrees. So sometimes there may be times you may be feeling like you are doing a lot of sacrifices in your life. You are only studying all the time. So, but let me read something for this. On this, let me read it. So waking up sometimes in the middle of the night, once you will get your certificates, once you will get your degrees, you will be on call, you will get phone calls from the patients in the hospital in the middle of the night. At 3 a.m. you will get a call that your patient is not well and you have to visit your patient in the hospital. It will be very routine for you, right? For all doctors, it's very routine. So waking up in the middle of the night, sacrificing sleep, leaving bed early, skipping TV shows, turning off Wi-Fi, stopping music, cancelling the plans, you will feel very low. No, it's not worth. But please believe me, don't worry. One day you will feel proud for doing all these things. One day you will be a doctor, a certified doctor, right? A doctor is a superhero for the patients. You are going to become someone's hope one day. You are going to become someone's hero one day. Come on. You are born to save lives. Right, so don't forget this and always be motivated, be positive while studying. Don't feel low that you are doing a lot of sacrifices. It's worth. I'm not saying it's easy, but I'm saying it's worth. It's worth. Thank you so much. Bye-bye.